Have you ever stopped to think about how we protect our food supply from, well, from invisible enemies? Today, we're going to dive deep into this amazing biological technique. It's kind of like a microscopic rescue mission for the plants we rely on every single day. It's called Meristem culture. And believe me, it's a totally fascinating look at how we keep our food safe.
Okay, let's kick things off with a pretty shocking number, 100%. Yeah, you heard that right. That's how much of a crop like bananas or apples can be completely wiped out by just one plant virus. We're talking total devastation. This isn't just a small problem. It's a huge threat to food security all over the world.
So, this invisible enemy, it's honestly one of the most destructive things in all of agriculture. I mean, we're dealing with over 2,000 different types of plant viruses. And here's the really scary part. With bugs or fungi, you can often spray something. But with a virus, once a plant is infected, that's usually it. There's no cure. It just spreads and spreads. And look at the scale of this problem. The numbers on this table are just, wow. We're not talking about niche crops here. These are staple foods for millions. Banana bunchy top virus? 100% loss. That's everything. And potatoes, a major food source for so many people, can lose over half their yield. This isn't some abstract problem for farmers. This directly affects the food that ends up on all of our plates. So you might be wondering, why are some plants so vulnerable? Well, it really boils down to how we grow them. A lot of our key crops, think potatoes, bananas, garlic, are grown vegetatively. That just means we're essentially making clones. Instead of planting a seed, you plant a piece of the parent plant. And because there are no seeds, there's no genetic lottery, no chance to mix things up and develop resistance. So any virus in the mother plant just gets passed right down to its clone, generation after generation. The viral load just builds and builds, and the whole variety gets weaker and weaker. Okay, but here's where the story takes a really cool turn. What if I told you that even inside a plant that's totally riddled with a virus, there's a tiny, perfect, pristine little sanctuary where the viruses just can't get in? It's like the plant has a secret weapon, a hidden safe zone. Let's go find it. And here it is. This is what we're looking for. This diagram points us right to it. See that? At the very, very tip of a growing shoot, there's this little dome of tissue. It's called the apical meristem. And when I say tiny, I mean tiny. We're talking less than a millimeter. But that little speck holds the entire key to growing a brand new, perfectly healthy plant. So what's the magic here? What makes this one tiny spot so special? Well, it's basically two things. First, the cells there are dividing like crazy, just super, super fast. So fast, in fact, that the virus literally can't keep up with the new growth. And second, this area doesn't have a developed vascular system. Think of the vascular system as the plant's internal highway, which viruses use to get around. Well, the meristem is like a brand new neighborhood with no roads built to it yet. So it basically outruns the infection and it's disconnected from the viral superhighway. Pretty clever, right? This discovery, which happened way back in the 1950s, was a complete game changer for agriculture. And as this quote says, it's become this hugely popular technique all around the world for one simple reason. It lets us grow plants that are totally free from pathogens, especially those nasty viruses. It's the solution for all those vulnerable crops we were just talking about. Okay, so we found the secret virus-free zone. Now, how do we actually use it? Well, for that, we have to head into the lab, where scientists perform what is basically a delicate, high-stakes rescue mission to grow a whole new plant from that one tiny piece of tissue. And this process, it's incredibly meticulous. Let's walk through it. First, you pick a good-looking shoot tip from the mother plant and get it totally sterilized. Then comes the tricky part. Under a microscope, a scientist has to carefully cut out that tiny little meristem. They call that piece the X-plant. They pop it onto a special nutrient gel, kind of like plant jello, to get it to start growing. Once it forms new shoots, they can multiply those, then move them to another medium to encourage roots to grow. And finally, once you have a whole little plantlet, it has to be slowly, carefully acclimatized to survive in the real world, outside the test tube. And these pictures give you a real sense of what this looks like in the lab. It's not happening on some garden bench. Everything has to be perfectly sterile inside a special hood using these tiny, precise tools. You can see the microscope they need just to see what they're doing, and there's the moment they place that little meristem into its new home. It's definitely high-tech gardening, that's for sure. Now, there's a really interesting challenge here, a delicate trade-off. The rule is, the smaller the piece of tissue you take, the better your chances are that it's 100% virus-free. But the smaller it is, the harder it is to keep it alive and get it to grow. So scientists are always trying to find that perfect sweet spot, small enough to be clean, but just big enough to survive. So Meristem culture is already pretty amazing on its own. But, some viruses are just incredibly stubborn. So to fight back, scientists have figured out ways to basically supercharge the whole process. They can combine it with other treatments to really crank up the success rate. This flowchart really shows how they stack the deck in their favor. The idea is to hit the virus before they even take the Meristem out. They can use things like thermotherapy, which is basically heat treatment, or chemotherapy with antiviral drugs. They can even use cryotherapy, which is extreme cold, or electrotherapy. The goal of all of these is to weaken the virus and knock it back, which gives the Meristem culture an even better shot at working perfectly. So, does this combo approach actually work? Oh yeah, the results are super clear. Using Meristem culture by itself is good, but, the success can be a bit hit or miss depending on the plant and the virus. But when you bring in these other therapies, you seriously boost the elimination rate. We're talking getting it up to 100% virus-free, even in really tough cases like garlic and potato. Let's zoom in on thermotherapy for a second, because it's one of the most common ones. The whole idea is pretty simple, actually. You take the infected plant and you grow it in a really hot room, like 35 to 40 degrees Celsius, for a few weeks. It's not hot enough to kill the plant, but it gives the virus a really hard time. It slows down its replication, stops it from moving around so easily, and the end result is a bigger virus-free zone at the top. That gives scientists a much larger, cleaner target to work with. And really, this is the main takeaway. This technology is so much bigger than just saving a few sick plants. Meristem culture has become a true cornerstone of modern agriculture. It's what we use to produce millions of healthy plants, to store the genetics of important varieties for the future, and to make sure that farmers all over the globe can get their hands on clean, healthy, productive crops. And that really brings us to the big question, doesn't it? Meristem culture is this incredible tool, and we've been getting better at it for decades. But now, we're facing huge new challenges, right? Climate change, a global population that just keeps growing. It really makes you think, what's the next big idea? How else can we use biotechnology to protect the plants that, let's be honest, we all rely on to survive?